Thanks for inviting me. I'm happy to be here. When you started with the Monty Python quote, I was a little afraid you were going to use one from the Dead Parrot skit and say that our next speaker has gone to his reward. He has passed over. But I haven't. I am eager to talk about ethical issues, but only if ethical issues in CRISPR defined and genome editing broadly are defined very broadly. I try to avoid being called a bioethicist. I'm a law professor. My training is in law. I work in bioethics, among other things. I work more broadly in what I consider law and the biosciences. My focus tends to be more practical and pragmatic and less philosophical. I'm less worried about Kant and more worried about the Congress. So when I talk about bioethical issues, I'm really, I think, talking about ethical, legal, social, political, and practical issues. And there are going to be a lot of them. And I suspect some of you are thinking, yeah, that's nice. That's why there are lawyers and bioethicists and all those others. Why should I worry about it? Why should I worry about it? And there are two answers to that. One is, no scientist is an island complete in his or herself, but you're all connected to the rest of society and the universe. And what affects the future of the human species affects each of us as members of that species. But more importantly, perhaps, if things go wrong either because there are accidents or mistakes, or because regulatory agencies or legislatures or others do wrong things, it can stop your work. It can keep you from doing good science. And trying to make sure that you can do good science is one of the reasons you should be interested in at least this broader definition of ethics. Interested in, and I think, and I will make a push for this at the end, participating in this broader discussion. So what I thought I'd do is talk about three categories of issues. Feel free to interrupt me at any time. If you throw fruit or vegetables, I'd prefer it's red, so it matches my Stanford sweater. It's dangerous being here at the home of the enemy bears, but trees are bigger than bears. Feel free to interrupt with questions, though I hope to leave plenty of time for questions and further discussion at the end. Three categories of things I want to talk about. Human germline genome editing, human non-germline, human somatic cell genome editing, and non-human genome editing, whether germline or somatic. The first, of course, has gotten the most attention, in part because of the workshop in late January that the IGI sponsored, and what came out of that, in part because of work in China, both the embryo work and the earlier, less remembered but non-trivial monkey work, where genetically edited monkey embryos actually led to live births of monkeys. I actually don't think it's going to be the most important, for reasons I'll explain. The human somatic cell stuff, apart from the really important and really not very controversial gene therapy applications for treating diseases, has gotten less attention. I think it deserves a little more attention. The one I hope to convince you of is actually going to be the most important, is going to be the non-human. The most important and the most scary. But let's start with the human, because that's been getting the most attention from the national academies, from the newspapers, from the magazines, and from politicians, which should make you all nervous. The reason I don't think this is going to be as important, the reasons I don't think this is going to be as important as people fear, are several. The first, and I think the most important, is it's actually not legal most places. It is not legal in the United States. Now, there is no statute in the United States that says you cannot do human genome germline editing. But there is the Federal Food, Drug, and Cosmetics Act and the Public Health Service Act. And although it may seem odd, for lawyers, this is easy to swallow. The FDA would consider a genomically edited embryo as a drug or a biologic. Probably not a medical device, but almost certainly either a drug or a biological product. And to use a drug or a biological product, to newly introduce one, you need FDA permission. You need FDA permission even to do it on an experimental basis. That's called an IND, an investigative new drug exception. To do it on a clinical basis, to do it regularly, you need to get FDA approval through a new drug application or a biological license application. And FDA is not likely to rush to make human genome editing legal, in part for really good, understandable reasons about safety, which I'll go into more in a second, but also in part because although FDA is one of the less political agencies in the United States, it is not a non-political agency, and it pays attention when the Congress that writes its appropriations for the White House that can order it around gets interested in one thing or another. FDA is only the United States. How many of you work in the United States? So the rest of you, I presume, either don't work or don't work in the United States. Almost every country that you work in is going to have an equivalent to the FDA. They will be different in detail, but they will still exist. And in those countries, the FDA equivalents are also, I would argue, not likely to rush into approving making babies after human genome editing. In addition, lots of countries have expressed laws that say you can't do this. The UK, which is a really interesting example because it's been one of the leaders in pushing for interesting reproductive advances. The first IVF baby, Louise Brown, was born actually almost to the day 37 years ago, sometime in late July. I think maybe the 22nd, so we may have just missed her birthday. She was born in the UK, and that wasn't a coincidence. The UK regulates it, though, and in order to do something new and different, you have to get approval, sometimes statutory approval. So some of you may remember just earlier this year, Parliament voted to allow the use of mitochondrial transfer, at least on an experimental basis, in the UK. So different countries have different rules. The European countries tend to be more restrictive than the United States, than Australia, than the East Asian countries. Some countries have no rules, but almost all countries have an FDA equivalent. So it's not going to be legal anytime soon. That's one reason I'm not concerned about it yet very much. Second reason, you'd have to be absolutely crazy to try to make a baby this way, given the safety issues involved. Some of the safety issues we saw with the Chinese work on the triploid embryos, the mosaicism, the off-target approaches, some of them will be problems with CRISPR-Cas9 or whatever technology ultimately gets used. I feel a little reluctant to say this at the home of CRISPR-Cas9, or one of the homes of CRISPR-Cas9. I don't believe that it's going to be what's going to be used in 10 years or 20 years. I think it's enormously important, but as an inflection point. Before the Model T Ford in the United States, there were automobiles, but they were really expensive. They broke down all the time. Only rich people had them. You needed to hire a chauffeur who was also a mechanic to keep it going. There were probably 10,000 cars in California. Now there are 30 million cars in California. There are probably more cars in California than there are people, and that may actually be true. None of them, almost none of them, is a Model T Ford, but the Model T Ford was the inflection point. It took it from being expensive and unreliable and difficult and risky to something everybody could have. And pretty soon everybody did. Similarly, I think CRISPR-Cas9 will be looked back on not as the thing that will be used forever, but the inflection point that made genome editing cheaper, faster, better, more accurate, that put it in the hands, almost literally, of anybody with a couple of $100, a garage, and maybe an undergraduate degree in molecular biology or biochemistry. So to use those technologies to make babies, we know that there are limitations. We know that there are problems that happen as a result of the process. One thing we don't know, certainly with humans, is does the process itself have some unexpected effects with respect to embryonic development, different from the problems we know, the off-target problems, the mosaicism problems from incomplete activity? Are there other problems in which, unexpected problems, in ways in which it interferes with embryonic and fetal development? And we don't know that. And we won't know that unless or until somebody tries it. But without a lot of work, you would be crazy to do it. You would be crazy in part because, let's say, I have a nasty genetic disease, and Sangamo or someone else comes up with a way to try to treat me. It doesn't work, I die. I'd say I would be sad, but I'd be dead, so I probably wouldn't be sad. No, I would have passed on. I would have departed. I wouldn't be dead, but I wouldn't be sad, but I'd hope some people would be sad. But you know, I'm a competent, more or less, adult. I got a disease, I take a risk, it doesn't work out, it dies with me. If you make a baby using a new method and something goes wrong, that baby didn't volunteer. Of course, none of us volunteered to be born, but that baby certainly didn't volunteer to be born in an experimental way. And unlike cutting a few years off the life of an adult, you're potentially setting up some human for 70, 80, 90 years of terrible existence. That's a heavy burden. Moving to making babies is much more serious than treating things in living people. And the uncertainties are also very, very high. So it seems to me that it would take 10 to 20 years of good safety research, both in vitro research, looking at where you can watch the first 6 days at least of human embryonic development, watch those, lots of animal research, including for something this serious, although the problems are huge, I think you'd want to do great apes. I think you'd want to do chimps before you actually move to humans. Macaques probably aren't close enough, or at least close enough to give people comfort that you can safely make babies this way. And what happens if you do it and something goes wrong? Well, I have lots of brothers and sisters in the law who would be happy to sue you or talk to you. Congress and other legislatures would be very happy to jump on it and ban making babies and making babies using this. And one of the problems anytime the legislature gets involved, and I think, although it may be more true of the American legislature than others, I believe this probably transcends all countries. Once the legislature gets involved, you really have no idea what's going to come out the other end. You know, they may think that they're doing X, they end up doing Y, they end up doing not X. You don't want that to happen any more than you have to. So I think people would be crazy to make babies, to try to make babies using this anytime soon. That doesn't mean that there aren't crazy people out there. 7.3 billion of us, some non-trivial percentage are crazy. Even among scientists, there are a few crazy ones. I actually think the mad scientist stereotype is false in an important way. Most scientists actually can't afford to be too transgressive because they need grants and they need tenure and they need the Nobel Prize and they need all these other things. But somebody with an undergrad degree and a couple $100 in a garage cares much less about being transgressive. So the democratization of genome editing that comes from a better, cheaper, faster, easier technology makes control sort of harder. So I suspect somebody will try this in the next 5 or 10 years. I hope it's not a disaster. But if 10 kids are born as a result of germline genome editing in the next 10 years out of 7.3 billion of us, it's not going to transform the species. Longer term, it might. But that's really at least 10 to 20 years off, which for some of you is a blink of the eye, and for some of us may be beyond our time horizon. Assume it's not. I'm still not all that concerned. I am concerned, but not desperately concerned, for another reason. I don't think there is likely to be a huge market, a huge demand for germline genome editing, at least early on. The most plausible reason to do it, the most politically acceptable, the reason that would motivate parents most is to avoid a genetic disease. But almost all genetic diseases can be avoided by other methods, notably pre-implantation genetic diagnosis. How many of you know about PGD? Well, not all of you. Okay, so PGD, you take an embryo. Either at day 3, when it's 8 cells, you take 1 cell off. Increasingly at day 5, it's a blastocyst. You take 5 or 10 cells from the outer shell of the blastocyst, the trophectoderm. You then do genetic analysis on the cells you've taken off, and you say, aha, This embryo will get beta thalassemia. This embryo won't. This embryo would be a carrier for it, but would not actually have the disease itself. And the parents say, well, we want to move these. We want to transfer the healthy ones to a uterus for possible implantation and birth as a child. That sounds like science fiction to many people. The first baby born as a result of PGD is now 25 years old. It goes back to 1990. Last year, 5% of the IVF cycles in the United States, so roughly 9,000 cycles, involved PGD. Around 3,000 babies were born in the United States alone after PGD last year, and next year and the year after, and the percentage is only going up. None of them, happily, are born without an arm or a leg or a head. You know, the cells that are left make up for the cells that you take off. It's a well-established, safe procedure. At least it is no less safe than regular IVF, which is a little less safe, arguably, than normal conception, although that's tricky too, because the groups that use them are not always the same. But it's a well-established technology. And you can use it in order to avoid genetic diseases, or in order to pick boy or girl, or in order to get HLA-matched cord blood to treat somebody else in the family. All things that you might want to genome engineer in the germline, you can do without it. Now, it isn't a wonderful procedure, mainly because it requires IVF. And IVF is expensive, unpleasant, and dangerous. At least unpleasant and dangerous for the women. For the men, it's not so bad. No one has been hospitalized for sperm donation, as far as I know, unless they were caught donating it to the wrong woman at the wrong time. But 1% of women who go through egg harvest every year end up in the hospital. And every few years, a woman dies as a result. And every year, several women will lose their reproductive capacities as a result of ovarian hyperstimulation leading to bad things with the ovaries. It's not that dangerous. 160,000 American women go through it every year. A couple million in the world go through it, but it's expensive, it's unpleasant, and it's somewhat risky. So it's not, unless those things change, and I'm desperately trying to finish a book by August 1st that says those things will change, but that's a different talk. Unless those things change, there's a constraint on it. But if you really want to avoid having a baby with Huntington's disease, when one of the parents has the Huntington's allele, avoid having a baby with the Huntington's allele who will later get Huntington's disease, it's available. Why do you need genome editing? There are a couple of exceptions to that. What if you've got an autosomal dominant disease and one of the prospective parents is homozygous for the disease allele? The only allele he has or she has to pass on to the child is a disease allele. The good news, I guess, is I can't think of very many autosomal dominant diseases where if you're homozygous, you will both live long enough and be healthy enough to want to have kids. But there will be some, even with Huntington's disease. There's some homozygotes who live into their 20s before they get diagnosed, mainly on the shores of Lake Maracaibo in Venezuela, where Huntington's was first really tracked down to its chromosomal origin. And they may have kids. And if they want to have a kid that is genetically theirs, that doesn't have the disease, genome editing is their only way out. Another example, maybe a little more common, although I don't know, two parents with the same autosomal recessive disorder. Two parents with, say, cystic fibrosis, where 30 years ago, you wouldn't have lived long enough to be a parent. But now the life expectancy is in the 30s or 40s in the US. CF people might be plausibly thought to be likely to group with and marry other CF people. And if they wanted to have a kid without CF, that is their kid genetically, genome editing is the only way they can do it. A couple of wilder examples involved that are limited to either, you might not be able to get a boy, or you might not be able to get a girl with some of the sex-linked ones, but I think the autosomal dominant homozygote and the two people with the same autosomal recessive disorder are the most plausible. How many of those are there in the world who are old enough and healthy enough and desire to have kids? More than zero. I would guess well less than 1 million, probably a few thousand. They have a medical reason to use this. Nobody else really does. One other example, mitochondrial disease. But of course, with mitochondrial disease, it looks like mitochondrial transfer made, just as PGD is an alternative to genome editing for most genetic disease, mitochondrial transfer may turn out to be an alternative to genome editing for mitochondrial disease. So why do the new, unproven, tricky thing if there's something that's either proven, as with PGD, or much closer and plausibly, quite plausibly safe, as with mitochondrial transfer? So I don't think there'll be a huge demand of it for that. There may or may not be a demand for other things, like the couple that wants a blue-eyed kid, and they're both homozygous for dark eye color. How sympathetic are we? Probably not very. Does that translate into regulation? Does that translate into physicians unwilling to do it if the couple's willing to pay enough? Probably not. Or people who want their kid to be particularly fast or particularly strong, or they think they found an allele associated with good math ability and they want somebody who's particularly high in math. The enhancement issues might be an issue. Those, I think, are less likely to happen soon because the push for them won't be as strong, and frankly because we don't know very much about enhancing alleles. We know a lot of alleles and genes associated with intelligence. Unfortunately, what we know is if this allele is broken, you have very low intelligence. We know the pathological intelligence genes. We don't know anything effectively about the normal range in intelligence other than it's good to be born to parents who have a lot of money and give you a good upbringing and environment and nutrition. Other than that, we don't know very much about the enhancing things, and I think that will push it off. It won't push it off forever, but I don't think it's going to be a near-term issue. Plus, if in fact the somatic cell gets good enough, how much are you going to worry about doing it at the germline if you can then do it when the kid is 4 or 5 or 10 or 12, and you see other aspects of what the child may develop in? Arguably, for things that need to be done during fetal development, you'd have to do it germline or very close to germline, but how many of those will there be? So that's a second reason I'm a little less concerned. And then there's a third reason. People talk about changing the human gene pool, changing the human germline. Well, you know, if you use this to change a 47 CAG repeat Huntington gene, pathogenic, to a 22 CAG repeat Huntington gene, non-pathogenic, you're changing one allele found in the human species to a different allele found in the human species. You're changing the allele frequency, but you're not changing what alleles are in humans. You're just changing their frequency. We do that all the time. Anybody here a type 1 diabetic? Good for you. It's not a terrible disease, but it's not a good thing to have. Until 100 years ago, type 1 diabetics basically almost never reproduced because they died before they became of reproductive age. Onset's typically in ages 5 to 15 or so, and it was untreatable. People died within a couple of weeks of being diagnosed. There is no powerful gene for type 1 diabetes. It's not a Mendelian disorder, but there are HLA haplotypes that are strongly associated that increase your risk 1 and maybe even 2 orders of magnitude for getting diabetes, type 1 diabetes. In the 1920s, 2 Canadians, Best and Banting, discovered insulin, discovered how to use insulin to keep type 1 diabetics alive. I don't think anybody's done the work. I don't know how you would do it, actually, but I'm willing to bet there are a lot more people in countries with good health care who have the high-risk alleles, the high-risk HLA haplotypes for type 1 diabetes than there were 100 years ago. Because now those people can reproduce, and they do. Medicine has made the gene pool worse. Well, kind of, sort of. But of course, natural selection is not just an issue of genes, but of environment. You change the environment, you change the selective pressures, you change the allele frequencies. Farming has changed our allele frequencies. Farming is different from hunter-gatherer. If you look at populations that have a long history of agriculture, they have different alleles in some of their genes, some that seem to improve their ability to digest starches, compared to populations with long histories of hunter-gatherer. Civilization changes our gene frequencies. If this is only being used to change gene frequencies within the human species, allele frequencies, what's the big deal? If you want to move chimpanzee genes into humans, chimpanzee alleles into humans, well, maybe that's an issue. If you want to move brand new synthetic alleles that George Church dreams up, or Lee Silver, even worse, into humans, that might be a big deal. But those aren't likely to be the uses, at least not anytime soon. If this actually happened, it would be mainly to change allele frequencies. I don't think it's that big a deal. So for all those reasons, I'm less worried about germline genome editing than most people are. Oh, to go back to that nice quote from Condorcet, or actually it was really your commentary on Condorcet. I'm a little surprised Condorcet said that given the collapse of classical civilization and the 1,000 years of the Middle Ages, when it appeared that Western Europe at least went backwards. But we can't ask him why he said that now. We could ask him, we're not getting an answer. The press loves scare stories. So do many bioethicists, so do many of our fellow citizens. Why? Because they're dramatic. Utopias are not very dramatic. Dystopias are dramatic. How do you write a novel about, here's this important change in molecular biology that makes everything better? You can write it, it's unlikely to sell very well. Brave New World, Gattaca, those sell a lot better. We like drama, we like tension. We like horror movies. Why do we like horror movies? It's that combination of fear and interest, but knowing you're safe. So the press, the public will always be more interested in the negative stories than the positive stories, at least at a general matter. The particular parts of the public who are positively affected by the stories, the disease families, they'll be excited and interested. But the general press is likely to always pull for the dramatic stuff because it's dramatic and we like drama. Yeah.
So in the case of diabetes versus
Yeah. Yeah.
what about the argument that one requires intervention with healthcare that's skyrocketing and
Yeah, you could say that we should just kill off everybody who's going to require a lot of healthcare resources. I would not make that argument publicly if I were you. But
people do that argument, right?
they do sometimes, although it's a brave soul who says we shouldn't take care of really sick infants because they're going to be a drain on our budget for the next 70 years. We should just let them die. Some people will say that. Not many. It's not going to be a popular position. And unless you're in a culture that is either very unusual, ancient Sparta might come to mind, or that is really, really pressed for funding its healthcare system, I don't think it's going to be a winner. But you could make that argument. In a way, though, it's sort of a... So remember, selection is a combination of the alleles and the environment. What you're saying, I think, is, well, we can do an environment that makes these alleles relatively benign, but it's too expensive. We don't want to change the environment that way. And at some point, we do that. There are, what would be an example? This isn't a great example, but there's a genetic variation that's associated with a really bad reaction to the element beryllium. Berylliosis is a really nasty disease. But it mainly affects people who have a particular genetic variation in the berylliosis gene. I don't know the backstory on it. We could try to eliminate beryllium from everywhere to protect those people. We're not going to spend that sort of money. Happily, beryllium is not so common, except actually in nuclear weapons plants. So it's a big problem. It was a big problem in the US nuclear weapons plant in Amarillo, Texas. We don't clean it up to affect everybody. So there are places where we do decide, we're just going to live with the disease or let people die because it's too expensive for us to change it. I don't think it's going to be a general argument. I thought you were going to go someplace else, though. What happens to all those diabetics and all those people with the type 1 diabetic HLA allele if civilization goes to hell and there's no longer modern medicine? And the answer is they die. When the environment changes, selective pressures change. Of course, should that happen, lots of other people will die too, and the overpopulation problem will not be a problem. But it is always this interplay between the alleles and the environment, and what I think we don't pay enough attention to is how much we make the environment. And we make choices that make the environment. Okay, so germline genome editing, reasons why I don't think it's a big deal. Why is it a big deal? Because it sounds really scary. Because if you play out some of the enhancement uses in particular, it is kind of scary to people. The changing the eye colors seems kind of frivolous, although people won't be happy about it. Increasing intelligence worries people. Moving foreign non-human or synthetic alleles into humans worries people. I think all that will be a worry, but probably not for a long time. There will probably be some regulation. The question will be trying to keep the regulation to regulation that avoids the things that are either bad or that people deeply fear, while preserving the things that are useful. And that requires educating science journalists, members of Congress, judges, and others, as well as citizens, about what the realities are, what can be done and what can't be done. So, germline editing. Questions on germline editing, because I want to move on to somatic cell. Okay, somatic cell. So, nobody's against curing, well, no one except those with the strongest concern about healthcare budgets is against curing genetic diseases in people who have been born. And even there, gene therapy looks like it's going to be cheap compared to all the anti-cancer monoclonal antibodies that are being churned out that help 2% of people with cancer and cost $1 million a year, but really help the bottom line of various local companies. Not to mention Genentech, exactly, but that's a popular thing. And it obviously has issues around safety and efficacy. Those are appropriate issues. There are also issues we deal with in all sorts of drugs and biologicals. They're not new issues. With gene therapy, they're a little more charged because we've had 35 years of frustration in establishing safety and efficacy. Frustration coupled with the occasional disaster, whether it's Jesse Gelsinger's death from gene therapy for OTC, or whether it's the sad results for the first gene therapy that really cured people, the SCID-X, boy in the bubble disease, where it cured kids and then gave a fair percentage of them leukemia, which killed some of them. Gene therapy's had a frustrating go, but I actually think that it's at the edge of becoming quite real. Europe just approved its first gene therapy product last year, and I suspect the US will approve one fairly soon. And 35 years certainly feels like it's a long chunk of anybody's career. But monoclonal antibodies took 25 or 30 years to get to market. It's not a real surprise. The issue with somatic cell is going to be the enhancement issue. It's going to be adults or kids having their somatic cells changed to try to make them not healthier, because that's not offensive to people, but somehow better. And maybe the best place to think about that, books have already been written on this, although it's not clear that anybody's done it, is sports enhancement, gene doping. It's a good new book called The Sports Gene by a guy named David Epstein. He's not sure that no one has tried it, but he's certainly not sure that anyone has tried it. To do things like add more copies of the erythropoietin gene, or add promoters, or try to add muscle mass genes, etc. That will concern people. And there are safety and efficacy issues. To the extent it is underground, those safety and efficacy issues become harder to enforce. So if you make it illegal, that's one thing, but if you make it illegal, then how do you know what's going on in the illegal centers? That's always a problem, as we know from drugs of abuse, and actually from sports doping right now with medications. It's not that it doesn't go on. We just don't know very much about it because nobody will talk about it because it gets them in trouble. So I think that's something to watch for. And I think actually sports may be the place where it comes first, because although we actually don't know whether any of these would really work, They're plausible stories. And people in sports, some of them will do almost anything to get that little improvement, which may make a tenfold change in their income or their fame. Or don't focus just on professionals. You know, the amateur runner out there, the guy who runs a marathon in 4 minutes, who wants to get down to 4 hours. The guy who runs a marathon in 4 minutes is not an amateur. The guy who runs it in 4 hours who wants to get down to 3.5, which still will get him, you know, the record is like 2 hours and 5 minutes. He's still not a serious runner. He may do a lot of things because he really cares about it. So that's something to watch. And trying to figure out with something that has the potential for going underground, how to regulate it is a really complicated matter. And once again, I come back to what I think is the big deal about CRISPR-Cas9 as a genome editing method. To the extent it makes it cheaper, faster, easier, more accurate, you put it in potentially in more hands. And so it's like meth. You know, if you needed a chemical company to synthesize meth, there wouldn't be as much meth, but methamphetamine. But once people figured out how to make it in their own kitchens or own labs and only blowing themselves up every 10 or 20% of the time, it becomes harder to control. Cheaper, faster, easier, more accurate genome editing means less controllable genome editing. Genome editing at Stanford or at Berkeley is going to be fairly well controlled, if not over-controlled. Genome editing in somebody's garage, who knows? That's all I want to say about the somatic cell stuff. I think that will be the issue to watch, and how we respond to it, and whether we can figure out a good way to respond to it, and whether it's even worth responding to. I mean, do we really care about athletes enhancing themselves? Since for a top flight athlete, there's really nothing other than genes that are unenhanced. You've got coaching, you've got equipment, you've got clothing, you've got sports psychology, you've got weightlifting, you've got nutritional supplements. The fastest natural runner in the world probably wouldn't win a high school track meet because of all the things that you need to, that you can learn and all the training you get, even at the high school level, that make you a better athlete. Is gene modification another step? That's a question for another day, but I think this will be the issue for somatic cell. Finally, the one I think is most important. Yeah?
Can I ask you a quick question?
Sure.
What about anti-aging therapies? That's a big market for that. And
Yeah.
also, there is correlations with DNA damage, even though you can't really envisage how you use current concepts.
Yeah, that's fair, and that's a nice example in a sense because it kind of straddles medical and non-medical. An actual anti-aging therapy could be sold through the medical system, could be produced by pharma or biotech, could go through FDA. We know, though, that lots of people do desperate things to try to keep themselves going. You know, human growth hormone is illegal in the United States for anything but a small number of cases. Almost everything else in the US, if it's approved by the FDA for one purpose, you can use it for, a doctor can use it for any purpose. It's called the off-label use exception. Congress passed a law to say no off-label use with HGH, human growth hormone. And yet you can buy nutritional supplements that are constantly claiming to increase your HGH, almost certainly fraudulently. But there's a black market for HGH. So I think the anti-aging stuff is a nice, interesting example because it kind of straddles legitimate medical and the potential for a black market use. The question would be, do you have a good, plausible, safe, and effective target? Maybe. And Calico and Larry Ellison and a bunch of other very rich Silicon Valley people hope so. I think if you get a good target, then it's something to worry about. I mean, something like telomerase to increase telomere length is yeah. Although with the mitochondrial DNA damage, I'd worry more about people, about sort of trying to do mitochondrial transfer, although to do it on a living adult person with 100 trillion cells is going to be a problem.
That's the scary part, right? Because you have to use very powerful.
Right. So I think I should add that to my list of most plausible enhancements, the anti-aging stuff. But it is scary because, you know, maybe telomere shortening leads to senescence and early death, although It's plausible. We don't know that yet. We do know that cells that make a lot of telomerase and that have long telomeres are often found in malignant tumors. So the potential for negative effects would be strong. But thank you. I will add that to my list of possible examples. The anti-aging therapies, I think, are another area where there may be there's enough desire that there may be a black market that moves into it sooner than it should.
Yeah, well, one more thing I want to say.
Just before you do, because my short-term memory is getting fried as I get older. It's not just a black market, like stem cell therapies, right? There are basically no stem cell therapies for for induced pluripotent stem cells or human embryonic stem cells that are FDA approved. There are a bunch of clinics that will sell you stem cell therapies. Most of them are in Mexico or Brazil or other places. So maybe in Western Europe, the United States, East Asia, these things will be regulated and limited so they're only used when safe and effective. But there will be countries that will allow their use, particularly if they're only used on rich foreigners. Go ahead.
I just wanted to add this information that actually even with cancer research, mitochondria have now become a very prominent target. It's kind of shifting away from basic mechanisms of cell growth, such as telomeres and stuff. It's more applicable things that we see reverse mitochondrial damage and you see remission in cancer. It's becoming really
right. I'm aware of some of that work. I'm not sure. I think it'll take 5 years before I begin to believe it. I've been around long enough that you see these fads come. Most of them come and go. Some of them come and stay. But at the early stages of them, you never really know. That's one of the concerns about mitochondrial transfer. Right now, there are a small number of really clearly, highly penetrant, mitochondrially based diseases. But there are a lot of people arguing there are all these other things. So the possibility of indication creep, of what starts out as a very small market getting to be a bigger market, even past the proof for that bigger market, in part because of patients who are desperate to do something and people who are happy to sell at a high profit rate things to patients who are desperate to do something coming up. So let's talk about mitochondria again in 5 years. Okay. But you know, it may turn out. Yeah.
I thought probably obesity or weight gain.
Careful.
That would become very kind of commercial.
Yeah. Again, but you need a nice target for genome editing, and one that was better than, say, stomach reduction surgery, which again is not an entirely benign procedure, but is safe and effective to a point. Yeah.
Because I have no professional background in law or ethics beyond the standard class of protection of human subjects and research, I'd love your informed professional perspective on enhancement in the following context.
Okay.
By the way, let me just subscribe my name to every single syllable of what you said about germline editing. Thank you. I cannot, as a practicing gene therapist, I cannot agree with you more strongly about everything you said there. For the enhancement bit, consider a plausible technical scenario. Across the bay at UCSF, they discovered that loss of function mutations in DEP2 allow people to sleep 6 hours a day with no apparent adverse effect. We and others are working on viral deliveries to the brain that penetrate the blood-brain barrier.
Mm-hmm.
So 10 years from now, there's a nuclease, Cas49, that as you correctly pointed out, we have the Ford Model T, which can be many colors, one that is black.
Right.
10 years from now, who knows? So
It could be gray.
So there's a viral serotype that intravenously administered routes to the substructure of the brain and gives people 6 hours of sleep. Ethical, sort of in terms of the Venn diagram of ethics and the law.
Okay.
Where will that even live?
So two different questions, ethics and law, and I'll take them in reverse order. On the legal side, although it has been rarely called upon to do it, FDA's jurisdiction applies to enhancement, not just to disease. The definition of a drug is an article that is used for diagnosis, treatment, cure, prevention of a disease or condition, or that affects the structure or function of the body of man or any other animal. And the structure function side covers everything that's not a food or a device. So FDA could regulate it. FDA hasn't really regulated a clearly enhancing biomedical technology yet, except a little bit. I mean, things like colored contact lenses are medical devices. They're almost always used only for, quote, enhancement to change somebody's eye color, but they're regulated as devices. Now, that's probably because soft contact lenses that they grew out of were regulated as devices. But I think that'll be an interesting issue to see if FDA steps up to the plate and regulates safety and efficacy of enhancing technologies. And we're on the edge of it. I also work on neuroscience, things like transcranial direct current stimulation. There's a company here called Think, T-H-Y-N-C. I hate cutesy spellings. That is selling a device that sends electricity through your skull, either to create a relax vibe or an energy vibe. What will FDA do about it? Not entirely clear. So that's the law side. And that gets even tougher in other countries where the details of the regulatory authority and jurisdiction are going to be different, as well as the cultures. I can imagine in some cultures enhancement being viewed more negatively, and in other cultures it being viewed more positively, and that being reflected in the legal setting. The ethical issues, I'm kind of an outlier here, I have to say. I think enhancement is what we do. I think the human species has been enhancing itself forever. Reading and writing, the greatest cognitive enhancer ever in terms of being able to convey, to increase the space and time over which you can convey information, and it changed the world. It changed our brains. There's an area back here known as the letterbox region, which is where people who read anything, not just alphabet languages, but character languages, they've processed the reading here. That wasn't involved for that. Our Pleistocene ancestors didn't read and write. Our brains have been changed by that enhancement. I think we do enhancement. And I think fears about to have somebody, to have a bioethicist fly across the country, go into an underground auditorium that's artificially lit, use PowerPoint and a microphone, and decry enhancement just really bothers me. You know, unless his arms are really tired from flying across the country. But I'm a bit of an outlier there. I do think there are three issues to worry about with any kinds of enhancement. One is safety and efficacy, which is fundamentally is an ethical issue. People don't think about it as an ethical issue, but it is unethical to sell something or provide something that's unsafe or ineffective. So safety and efficacy, the biggest. Fairness is another issue. But fairness doesn't necessarily mean ban everything. Maybe it means make it available to everybody. And even that depends on the nature of the enhancement. I'm going to use brain stuff because that's the area where I've written most on it. We like to think that enhancement might make everybody, say, 50 points better. But maybe there's a cap, and it just moves people closer to a natural cap, in which case it would be a pro-fairness thing because it would bring lower people up disproportionately. But fairness is a legitimate, real issue. It's just there are a lot of different potential ways it plays out. And then coercion, being forced to use it. I am not a fan of the naturalness argument, that it's unnatural. I think humans are unnatural. Either humans are unnatural or we're part of nature and everything we do is natural. The argument that I find a little trickier is an argument about identity and whether you're changing your identity in ways, but I also think identities are really fluid. And everything we do changes our identity as well. And then there's it's cheating argument, which for various reasons I'm not keen on. So that's my take on enhancement, but I am more pro-enhancement than a lot of people are. So, non-humans, can we go to bugs and other creepy crawlies? CRISPR critters. Somebody, I wish I could claim that. I heard somebody describe non-human uses of CRISPR as CRISPR critters, and I can't remember who it is. I'd love to give them attribution, because I think it's just such a great term, CRISPR critters. I think this is the real issue, because non-humans are a hell of a lot easier to work on than humans. Humans are terrible lab animals. You know, we got lawyers, we got long generation times, we don't follow orders, we lie. Rats are a lot easier. Fruit flies are easier still. And more importantly, we've got lawyers and they don't. And that means safety is a much smaller issue. If you do something to fruit flies that means 99% of the fruit fly babies don't survive, no one's very upset. Not even, I suspect, the fruit fly mamas. If you do it to humans, you're in prison for a long time. So dealing with non-humans is a lot easier for a whole bunch of reasons, and it's tempting because you could do some really good things. I suspect in this audience, although some of you may be from Europe, so I may be wrong, that there aren't a lot of anti-GM crop or food people here. GM foods, GM crops do a lot of good things. You could do them faster and easier and better, presumably, as genome editing gets better. That's good. Being able to make biofuels by enhancing the ability of algae to make hydrocarbons that are easily turned into something else. Good potential use. Bioremediation. Figure out a way to edit the genome of a bacteria that lives in highly acidic soils so it picks up heavy metals. And then you can harvest the bacteria and you can do bioremediation. People are doing this at some level. The other quote I liked, I love your Sidney Brenner quote. Scientific revolutions, I think, are mainly revolutions in tools. They're usually physical tools. Sometimes they're conceptual tools like calculus, but they're usually physical tools. Genome editing becomes more revolutionary as it becomes cheaper, easier, and faster, like the Model T. So bioremediation, big jump. All those are good things. Probably. But what happens if instead of being done by a company that's regulated by a government agency, it's done by a 23-year-old in his garage who thinks, boy, it would be really cool, wouldn't it, to change mosquitoes so that they don't bite humans. And here's a way I think I can do it. And does it and releases them. And you guys, have you dealt with gene drive at all? Puts gene drive in so it spreads through the population quickly. He may end up changing all of that species of mosquito in his county, in his state, in the world, with nobody looking at the side effects, the potential downsides. I'm not a big fan of mosquitoes. They probably do something useful. But changing them without regulation is worrisome. So even good kinds of changes. The scariest one, of course, E. coli. Found in lots of labs, found in all our guts, lots of different strains. A couple of the strains are really nasty and cause severe human illness. Their genomes are published. The lab strain's genome is published. How hard would it be to use genome editing to change the lab strain of E. coli into a pathogenic strain of E. coli and then release it? Let alone cowpox to smallpox or mousepox to smallpox. The genomes are known. To the extent genome editing gets easier and cheaper and faster, biological warfare issues become much worse. I wish I had a solution for that. I don't have a clue how we're going to deal with that, because I think that's going to be a real problem. The only good news is biowar has some inherent constraints in that it's hard to avoid blowback, the biology, the weapons hurting your own population or your own soldiers, and it's harder to control. But I do think that improvements in genome editing lead to increased capability for biowarfare. Yeah.
So I'd like to hear your thoughts. There was this controversy last year, 2 years ago, about releasing this particular strain of influenza that they'd identified a couple of really pathogenic parts.
Releasing the data about, the information about, not the strain. At least if it was about the strain, they didn't tell us.
I think that may have been a slip of the tongue there. What are you actually thinking about?
Not great.
Do we still have Freudian slips post-Freud?
So, yeah, what is the balance? How do you get the data to other scientists that are truly trying to do good and keep out of the hands of the...
I think it's a real hard problem. I mean, there are many problems that are hard just because they're complicated. Figuring out how many seconds since Julius Caesar was assassinated is complicated, but it's not hard. There are other problems that are politically hard. Dealing with water in California in drought years actually isn't hard except politically. It's really hard. But then there are problems that are just really hard problems because competing principles that are both valuable are at inherent conflict. So wanting to protect people, wanting to keep security is a valid, appropriate principle. Wanting scientific openness so people can learn from each other is another valid principle. And those problems you never solve. You just try to manage them. And the management of those problems almost always leads to something that people will not be entirely happy with. And I'm tempted to say something about marriages, but the compromises involved in a marriage. Rarely are a couple entirely in sync on everything, but they usually work it out, live with each other, make compromises. Hard problems. And I don't know the answer to that. On that particular issue, I am more in tune with my Stanford colleague David Relman and the dissident group that then got fired from the National Bioscience Advisory Board, or whatever it was called, because they wanted more restrictions in that particular research. But it all depends on the particular circumstances.
I don't think complex problems we don't typically have very simple.
Yeah, well, they always have. H.L. Mencken said, every difficult problem has a solution that's simple, straightforward, and wrong. I didn't get that quite right, but I think it was Mencken, yeah.
I don't want to take too much time. But first, until you've just said this, I never thought about this. Somebody was one of the parents of editing. It's kind of sad. So I think that I've actually never given some thought to the fact that by the democratization of editing that we've enabled democratizes it hard.
Right.
And raw.
And democratization is not an entirely benign process.
Do you think that education can be raw? In other words,
Sure.
is this something that we can start telling people? Like, folks, as it becomes easier and easier to tweak the genome as CRISPR fritters become reality, please understand, and so on and so on. Is there
Certainly there's a role for education. It doesn't always work out the way one hopes. So to go back to GM foods, there's some interesting, kind of surprising studies in the US that show that the more science literate people are, the more opposed they are to GM food, or the more interested they are in mandatory labeling of GM food. It's I think, an important step, but it's not a panacea. And the other thing that education does is give some people who might have bad motives some good ideas. Ideas that from their perspective are good ideas. So yes, education should be pursued, but you shouldn't count on it working. Even if, how many of you have had teenage kids? How many of you were teenage kids, okay? Education doesn't always sink in. Even if you could get past that, you still have problems of how well it would work out. Yeah?
You brought up weaponization.
Yeah.
In terms of non-human editing.
Yeah, you could do the human stuff too.
Right, is there any discussion, especially policy where they discuss scenarios where either germ-like or somatic cell editing occurs, makes weaponized and target specific, say, ethnic?
So the nightmare of ethnic-specific targeting goes back at least 20 years. I cut my teeth in the genome business working on the not entirely successful, largely failed Human Genome Diversity Project. And in 1995, I was in Quetzaltenango, Guatemala, at the conference of the World Council of Indigenous Peoples, where I was widely accused of being a CIA agent. Because, of course, Luca Cavalli-Sforza and Mark Feldman and all these other, Mary Claire King, they wanted indigenous people's DNA so the CIA could make weapons to kill them off. And, frankly, given what's happened to indigenous peoples, especially in the Americas, I think a sense of paranoia is probably a survival trait. The tribes that weren't paranoid aren't around anymore. But it was frustrating. I think human somatic cell genome editing for weapons purposes is plausible. I've not heard it discussed very seriously. I have heard a little bit about a way to, I think it may have been tried in mice to induce lung cancers using genome editing. If it's being talked about by the American military and intelligence agencies, it's a level where they would kill me if I found out about it. But I suspect it is. I hope mainly as a defensive question. How do we defend against it? The ethnic targeting is harder, in part because, with very rare exceptions, there's nothing that is unique to one particular ethnic group. We're all sort of blurs. but certainly you could do something that might affect 80% of one group and only 20% of most others, except for the ones who live near the one group, where it's 50%, et cetera. It's like Rh negative. The population in the world with the highest level of Rh negative is the Basques, who live in the Pyrenees between France and Spain. Most of the rest of the world has very low Rh negative levels. The two populations with the second highest level of Rh negative are the southern French and the northern Spanish, the people who live near the Basques. So targeting would be difficult. That's a serious nightmare.
Well, perhaps a better example is HIV. I've heard these conspiracy theories that
Right.
are targeted towards the gay population.
Or was targeted toward the Haitians or the Africans or the hemophiliacs or, yep. And, you know, you want to say that's crazy and paranoid, but from some of those perspectives, it may not look so crazy and paranoid, depending on what kind of a history of oppression your people have had. So I think those are appropriate concerns, but like the weaponization of non-human things, I don't see a great answer for it. It's not near term, but it's worth thinking about. A few last thoughts on non-human. I actually think what's likely to create the greatest controversy, assuming there's no weaponization that actually gets used, that would clearly be a big deal. I think it'll be frivolous uses, sort of frivolous uses. I've written about one of these, and I can't quite call it frivolous, but I think it's maybe the most interesting thing I work on, but maybe the least important: de-extinction. We know the woolly mammoth genome. We know the Asian elephant genome. George Church is already modifying a cell line of Asian elephants, an Asian elephant cell line, to make them more like mammoths. We're not going to get dinosaurs. It won't be Jurassic Park because the DNA doesn't hang around for 66.4 million years. But Pleistocene Park, I suspect some of the people in this room will live to see Pleistocene Park. And, you know, I'd pay to see woolly mammoths and saber-toothed cats. Oh, yeah. Giant ground sloths. Maybe not the cave bears from Berkeley, but. So I think we'll see that use.
Do you have an ethical issue?
See the article I wrote, the policy forum I co-wrote in Science in April of 2013 is the short answer. The longer answer is not a huge one. I think it needs to be regulated. You don't want to have it be this nasty invasive creature that you let out. Although frankly, our ancestors did a pretty good job of killing off mammoths with spears. I think we wouldn't have much trouble hunting down escaped mammoths if it turned out to be a problem. Passenger pigeons, an extinct species, you put pigeons out in the world, in the woods, hunting those down, that could be a real problem. Although our ancestors managed to kill them off too. That's one. Pets. How many of you have had a dog as a pet? Any of you have a really big dog? What kind?
God, I don't know the name in English. Sorry. It's like I don't know, a hound.
Okay, big dog. Saint Bernard or a Great Dane or an Irish Wolfhound. Anybody have a lunchbox dog? What kind?
Chihuahua.
Oh, God. And I slowly moved away from the woman with the Chihuahua. I know people like rats and, you know, okay. Irish Wolfhounds and Chihuahuas were wolves. In surprisingly few thousands of years, we turned them from actually kind of dog precursors. They spent a long time being sort of undifferentiated non-wolf dogs, but all the current breeds are just a few thousand years old, and most of them are only a couple hundred years old. We did that old-fashioned way. What could you do with CRISPR-Cas9? My guess is sometime in the next 20 years, some billionaire with a 12-year-old daughter is going to pay to have made a unicorn. Take a horse, figure out genes for one horn, maybe genes for two horns, and just cut the other one off. Got a unicorn. Somebody's going to make a dragon. Now, the dragon won't fly. Physics won't be repealed. And the dragon won't breathe fire, probably. And I hope it will be the relatively friendly, traditional East Asian dragon rather than the really evil, nasty European dragon, or the Hungarian horntail, or the Norwegian ridgeback for the Harry Potter fans. Somebody's going to do it. And that'll lead to an uproar. Last point, the ones I'm worrying most about, artists. Can't trust those damn artists. Seriously, scientists can't afford to be too transgressive usually, Because you need grants, and you need tenure, and you need facilities, and you need friendly IRBs and biosafety committees and funders. Artists, some of them at least, love to be transgressive because that's the way they get publicity. There's already this evolving school, evolving's the right word, of bioartists. One guy, Edouard Koch, put green fluorescent protein into a rabbit. I was actually sort of pleased that it didn't work. It did work, but it didn't get into the hair. So the only parts of the rabbit that glowed green in UV light were its nose, the greens of its eyes, and the pads on its feet. But people will try this more, and artists will do bizarre and outrageous things, because for some of them, that's art. What that means for us is when an artist does something bizarre and outrageous, it's gene editing, genome editing, and it stirs up the public against all uses, potentially all uses. Genome editing becomes bad because it's this thing that makes sick dragons or green rabbits or something else. And it's not a particularly logical objection. I don't know that there's any strong ethical argument against art that's green rabbits, but it's a pretty strong visceral objection. And this is where my law background, politics is part of ethics, or ethics at least, ethics and politics are interlinked, and politics will affect you wherever you're working. So, the ethical setting, broadly conceived, has some real problems, some real issues. I think the weaponization, biowarfare issues are real and are very hard, and I don't have a clue how to deal with them. And then it has a bunch of issues that aren't necessarily tangible or measurable, but have strong visceral effects on people, which have then political ramifications. If you want to be able to do your science, you need to have a supportive environment. One that funds your science, one that doesn't ban your science. And to do that, the people who are doing the funding and the regulating and so on need to understand your science somewhat. They don't need to understand all of it. They need to understand enough of it. They need to have confidence in you and what you're doing. They need to know scientists. Scientists need to talk to them. Most scientists I know hate talking to the press. I talk to the press all the time. And I can guarantee the only way not to get screwed over by the press is not to talk to them. And that even doesn't work, because one time in 10, they'll screw something up. Because they do. They're human. They don't understand it all that well. But if you don't talk to the press, who does? If good scientists don't talk to the press, bad scientists do. Talk to the press. Talk to high school groups. Talk to the local Rotary. Talk to people about what you do, why it's important, why it isn't as scary as they think it is, why it's important, and why it's important. And mainly why it's important for relieving human suffering. Because it is. And that's the issue people care most about. And if science doesn't communicate that, ideologues, demagogues, people who are looking for headlines and votes and visceral appeals and responses to visceral appeals will win this fight in various ways. And science, and ultimately I think humanity, will suffer as a result. So you now know as much about the ethics of human germline, of human and non-human, germline and somatic cell genome editing as all but probably a couple thousand people in the world. I'm not claiming that I'm all that great, but there just hasn't been many people talking about it for very long. You're in the 0.001% in terms of knowledge. Plus, you know the science as a result of the last 4.5 days. So don't sit on it. Feel I charge you. Because you have with ability comes responsibility. You have the ability to potentially make a difference in both how these techniques are used and how they're perceived and how they're regulated. With that ability comes the responsibility to use it and use it well.